Good day, everyone. Today, I present our research on differential expression profiles in breast cancer cell lines under hypoxia and normoxia conditions identified by Illumina sequencing. At the beginning, please allow me to give some general introduction. Hypoxia is a critical microenvironmental factor influencing the behavior of breast cancer cells through pathways such as HIF-1 alpha and VEGF signaling. This evidence suggesting that hypoxia significantly alters the expression of genes and microRNAs, leading to changes in tumor growth, invasion, and metastasis. This hypoxic environment can induce a more aggressive phenotype in breast cancer cells, making the study of these conditions highly relevant. Breast cancer cell lines under normoxic and hypoxic conditions exhibit distinct gene expression files. Studies indicate that these distinct profiles make these cell lines a valuable model for studying the molecular mechanisms underlying breast cancer progression. By understanding differential gene expression under these conditions, we can identify potential biomarkers for early detection and novel therapeutic targets for breast cancer treatment. For example, certain hypoxia-induced genes may serve as early indicators for tumor progression or as targets for therapeutic intervention. In Figure 1, we illustrate the regulatory mechanisms of HIF-1 alpha under normoxic and hypoxic conditions. The figure also highlights the genes that directly regulated by HIF-1 alpha, categorized by biological function. These genes include representatives involved in cell proliferation and survival, metabolism, invasion and metastasis, angiogenesis, pH regulation, and stem cell maintenance. Understanding this regulatory network provides insights into how hypoxia drives the malignant behavior of breast cell cancers. Our study aimed to utilize Illumina sequencing to identify differentially expressed genes and microRNAs in breast cancer cells lies under hypoxia and normoxia conditions. By comparing these conditions, we hope to uncover novel insights into molecular adaptation that cancer cells undergo in response to hypoxia. These insights gained from this research could provide new avenues for early diagnosis and more effective treatment strategies for breast cancer.
I will introduce you to the main methodologies parts of this study. In this study, they use three breast cancer cell lines MCF7, MD8, MD231, and T470. They calculate them under two oxygen conditions. Normal cells 21% oxygen and hyperoxia cells at 0.5 to 1% oxygen. First, they extract totally RNA from the cell using PRISOS reagents. The quality of the extracted RNA was assessed by an Agilent 2100 cell analyzer and non-neutral photometal. Only RNA sample of accessible qualities were used for sequencing library constructions. Next, they performed RNA sequencing using the Illumina HiScale 2500 platform. This data is very important because it generates a large amount of data for them to analyze. For data analysis, they use ERG and GraphPad primaries and Excel software. Those tools help them to analyze the data statistically and to ensure the reliability for the result. To further understand the function of the gene, they use the bioinformatic tools such as CLC Gene Workbench, MurrayBase, and MurrayNet. With those tools, they could read annotated and functionally analyze the sequences. Finally, they also interpret genes function by gene out knowledge and the case pathways, and verify change in gene expressing using qPCR.
而根 coming to the results, it was taken from an article by Ye. This talks about the intratumoral hypoxia, which is linked to cancer progression and treatment failure, highlighting the importance of understanding hypoxic responses. This was a large-scale RNA sequencing analysis with 31 breast cancer cell lines and normal mammary epithelial cells. It was under condition of control, which is 20% of oxygen, and hypoxic condition, which is 1% of oxygen. The first figure consists of two heat maps, A and B, showing gene expression values for a 42 gene hypoxia signature in 31 breast cancer cell lines. Panel A shows gene expression levels under standard tissue culture condition, which is 20% of oxygen, and panel B shows gene expression levels under combined conditions of 20% and 1% of oxygen conditions. Now, each row represents a gene and each column represents a breast cancer cell line. The color indicates the level of gene expression. So red indicates high and blue indicates low gene expression. Coming to the results, the basal-like breast cancer cells exhibit higher endogenous levels of the hypoxia signature genes compared to the other subtypes, which is HER2, luminal A, luminal B, and normal-like. This suggests that basal-like cells have a distinct hypoxic gene expression profile even under normoxic conditions. For the panel B, the hypoxic condition further amplifies the expression of the hypoxia signature genes, particularly in basal-like breast cancer cells. This suggests these cells are more responsive to low oxygen levels. Moving on to figure 2, it consists of bar graphs A and B showing the number of genes upregulated and downregulated in response to hypoxic conditions. Panel A shows the number of upregulated genes and panel B shows the number of downregulated genes. Now, each bar represents a breast cancer cell line and the height indicates the number of genes upregulated and downregulated under hypoxic conditions. The shading within the bars indicates the fold change levels. The panel A shows that basal-like cell lines, which are the leftmost bars, show the highest number of upregulated genes in response to hypoxia, indicating a strong transcriptional response to low oxygen levels. For panel B, the basal-like cell lines also show a significant number of downregulated genes under hypoxic conditions, indicating a substantial reprogramming of gene expression in response to hypoxia. Now, figure 3. This includes multiple panels, that is A to E, focusing on specific genes, which are Casp14, FUT11, DNAH11, TCA-F2, and their regulations under hypoxic conditions and the requirement of HIF-1 alpha for this regulation. Now, panel A shows a qPCR data of the expressions of Casp14, FUT11, DNAH11, and TCA-F2 in various breast cancer cells under normoxic and hypoxic condition for 24 hours. The bar represents relative gene expression levels normalized to a control. The results are that these genes are significantly upregulated under hypoxic conditions across different cell lines, suggesting they are part of the hypoxia response. Now, panel B is the qPCR data which shows the expression of the same genes in MCF7 or MDA-MB231 cell lines transfected but non-target control or CRISPR vectors targeting HIF-1 alpha. The bars show relative gene expression levels indicating the requirement of HIF-1 alpha for the hypoxic induction. In results, it showed that the knockdown of HIF-1 alpha significantly reduces the hypoxic induction of these genes, confirming their regulation is dependent on HIF-1 alpha. For panel C, the qPCR data showing the expression of DNAH11 and TCA-F2 in similar conditions as panel B. The results showed that consistent with the other genes, the hypoxic induction of DNAH11 and TCA-F2 is also dependent on HIF-1 alpha. Panel D is the schematic showing the 5 untranslated region of the human Casp14 gene and the locations of candidate HIF binding sites. It interprets that potential sites where HIF-1 alpha might bind to regulate Casp14 expression. The results provides a basis for further investigation into the direct binding of HIF-1 alpha to these sites. Lastly, panel E is the chromatin immunoprecipitation assay, which shows the binding of HIF-1 alpha, HIF-2 alpha, and HIF-1 beta to the candidate HIF binding sites in the Casp14 gene. The bars represent relative binding enrichment at 3 different sites. The results is that the significant binding of HIF-1 alpha to these sites under hypoxic conditions supporting the direct regulation of Casp14 by HIF-1 alpha. In conclusion, these results collectively suggest that basal-like breast cancer cells have a distinct and robust hypoxic gene expression profile, and the upregulation of certain hypoxia-responsive genes is heavily dependent on the presence of HIF-1 alpha. This indicates that HIF-1 alpha plays a crucial role in mediating the hypoxic responses in these cancer cells.
Hello, so I'll continue with the discussion part. So, as we have seen, while hypoxia-targeted drugs offer promise for improving cancer treatment, recent clinical trial failures necessitate a reassessment of approaches. These complex biology of hypoxia and unclear clinical indication posed significant challenges. Better patient selection criteria are reliable. Non-invasive diagnostic techniques are needed. A study on breast cancer cells lines highlight the significant transcriptional heterogeneity in under hypoxia and identifies novel HIF regulated genes that could serve as biomarkers. Particularly, basal-like breast cancer cells show intrusive HIF pathway activation, contributing to their aggressiveness and poor prognosis. Further research into these mechanisms is essential for developing an effective hypoxia-targeted therapies. Thank you.
Finally, it's here for me to do the conclusion of our research. Let's go ahead. Hypoxia is a critical factor that influences the behavior of breast cancer cells, leading to changes in gene and microRNA expression. This can result in tumor growth, invasion, and metastasis. Understanding these changes is essential for identifying potential biomarkers for early detection and novel therapeutic targets. Our study used 3 breast cancer cell lines, MCF7, MDA-MB231, and T47D, cultivated under normoxic with 21% of oxygen and hypoxic with 0.5 or 1% of oxygen conditions. We employed Illumina sequencing to identify differentially expressed genes and microRNAs under these conditions. The results reveal significant alterations in gene expression profiles between hypoxic and normoxic conditions. These findings provide valuable insights into the molecular adaptations of breast cancer cells to hypoxia, which could lead to new avenues for early diagnosis and more effective treatment strategies for breast cancer. So that's all for our poster presentation. Thank you, and many thanks to my group members.